Okay, hello everyone. I feel very honored to give the presentation today, and my name is Ting Qi. I'm a PhD student from Southeast University, State Key Laboratory of Digital Medical Engineering, and I major in bioinformatics, and my research directions includes liquid biopsy, cancer early screening, deep learning, and epigenetics. Today, I want to share my recent work named Prediction of Transcription Factor Binding Sites on cell-free DNA based on deep learning. And my report will cover the following contents. The first part is background. Cell-free DNA is the DNA fragments that are released into the plasma via apoptotic and nucleotic events which carry in the genetic and epigenetic information of its origin tissue. Like this. This is the SNP and indels, and this is the methylation information. This information in cell-free DNA can deduce its origin tissues. Therefore, based on these features, its potential applications include early screening and diagnosis of cancer, non-invasive prenatal diagnosis, and monitoring of organ transplant rejection. Our work focuses on the regulation mechanisms reflected by the cell-free DNA fragmentomics. Researches on cell-free DNA fragmentomics shows that cell-free DNA has the distribution of cell-free DNA on the genome is not random. It is related to the disease status, just like the picture. We can say this is the healthy individual and this is the cancer individual. They have different epigenomic regulations. And this information can be deduced by the cell-free DNA fragment patterns, and these fragment patterns can reflect the tissue-specific nucleosome footprints and expression regulations. Also, there are research on the cell-free DNA shows that short cell-free DNA fragmentomics are enriched in the transcription factor binding sites and exhibits higher coverage patterns compared to other regions, just like this picture. And this is the TFBS 1, 2, 3, and in this place, it has higher coverage patterns compared to its other regions, and this coverage pattern is related to the disease status. So it's very important to locate the TFBS position in the cell-free DNA, but the problem is that precisely locating TFBS on cell-free DNA is very challenging because at present there is no such ChIP-seq data, ChIP-seq experiment on cell-free DNA. Hence, we started from the perspective of sequence characteristics, because each TF has specific motif information. Based on the deep learning model to train the existing cell line ChIP-seq data to predict potential TFBS on cell-free DNA. And our blood-based localization of transcription factor binding sites has the potential to reflect the intrinsic regulatory mechanisms to find some specific biomarkers, and these biomarkers provide technical support for real-time monitoring of disease status in the clinical. And this is the framework of our research. First, we download the cell line chip-seq TF-BS sequence from the database. Then the sequence were one hot encode, just like this, one hot encode. Then this is the input metric. The metric were fed into the CNN plus LSTM model to training. Then we will get a prediction score. And we use this trained model, this pre-trained model, to predict cell-free DNA fragments that is less than 17 base pair to find some specific TF-BS. Then we analyze the TF-BS characteristics on cell-free DNA. Next, let's see the results. First, we trained 4 data sets, 2 cell line in 2 TF. That is 4 cell line. And the highest AUC value reaches 84%, just like this picture. And the lowest loss was reached when training to 20 epochs. Actually, I trained 40 as well, but as the epochs increase and the accuracy decrease, so I found 20 epochs is the best. Then I wonder if our model could detect the CTCF and FOXA1, the TF's motif. We use the as we all know, the convolution kernel of CNN is similar to PWM. That is position weight metric, and the list is like a window. You can go through the sequence to find the motif, and our results shows that the motif filters can accurately learn the motif information of the TF. Just like the picture, we can say this is the CTCF motif download from the Jasper database. And this is the CTCF motif learned from our model. And these two are almost the same. Then we use the pre-trained model to predict cell-free DNA TF-BS. We can see that the feeding curve of cell-free DNA fragments density and the model prediction score shows that the different sample, this is the 2 different samples, have the same peak in the same TF, like the CTCF and the FOXA1, and these 2 have the similar trade as well. And this phenomenon indicates that cell-free DNA could infer the binding length of TF in vivo. Then we analyzed the coverage depth around the TF-BFs. We found higher coverage depth like this occurred in cell-free DNA TF-BS compared to that in its upstream and downstream region. And this results is consistent with other people's published work. And we also found that the CTCF and FOXA1's motif can be predicted in cell-free DNA TF-BS. And this result is consistent with the previous download CTCF. These results together shows that cell-free DNA can be predicted based on the deep learning model. Next, I analyzed the distribution of cell-free DNA TF-BS on the genome and on the messenger RNA, and we found the different cell lines with the same transcription factor have similar distributions, just like the purple line and the blue line. They have the same trend and the same distributions around in the 3 UTR region, and the same situation in the bar plot. we can say this 2 bar and this 2 bar, they have the similar distributions. Also, we found more TFBS are distributed in the CTCF transcription factor than the FOXA1, and this is interesting results. Then we also analyzed the target genes from our method. We found the FOXA1 specific target genes like the ERBB2, ESR, PSA, and the CTCF specific target genes can be found in our the target gene list. And we also perform the KGG pathway analysis in these target genes. We found some interesting results, like the cancer-related pathways can be enriched in this cell line, and this cell line. And this cell line is the non-carcinoma cell line. Also, we found some pathways. These pathways are related to cell activities, and these pathways are related to signal transduction. Also, we found small cell non-cancer pathways can also be enriched in our cell lines, in our pathways. And these results indicate that self-DNA has the potential to reflect TF regulation non-invasively. Okay, let's make a conclusion about this work. First, our deep learning model can accurately identify sequence with TFBS that we are interested in, and this model can predict TFBS of cell-free DNA, and it has the potential to predict the binding sequence length in vivo. And this result is very important because when use the chip-seq experiment to detect the binding length of TF in the vivo, it is very difficult because we can get a region, not the specific length, but from the cell-free DNA fragment length, we can find the specific binding length. Last, predicting TFBS on cell-free DNA can reflect TF target genes and functional regulation. And my work has not finished yet. We plan to filter out some disease-specific target genes and predict the cancer monitoring. Then we want to verify our results from the experiment. That's all. Thanks for your attention.
Okay, thank you, Qi-Ting, for an interesting talk. Any questions from the audience? Are you deep sleeping? Oh, yep.
Thank you for the interesting talk. I'm working on a similar problem, and I just had a question regarding how you, what were the negative examples that you used in your training set?
Okay. You mean the negative samples, right?
Yes.
We use the sequence. We just select select 1/4 from the whole database. Then we use this as the testing as the test set.
No, I meant, sorry. So for example, so as I understand, your training set was composed of already existing TF chip-seq binding sites. So your label ones would be bound sites. So I was just wondering how you constructed your label zeros. Were they like random sites from the genome, or were they scrambled sites of the original chip-seq binding sites? Were they like scrambled sequences from the original chip-seq binding sites?
The chip-seq binding sites are the chip-seq binding sites are detected in the chip-seq cell line experiments, and these data are downloaded from GTRD database, and we just choose 4 cell lines, 2 cell line and 2 TF to training and test our model. Then we use the model to use the pre-trained model to predict the sequence, the motif characteristics on the cell-free DNA fragments. Try to find some potential TFBS on cell-free DNA fragments, and to get the potential target genes, and try to use these target genes to monitor disease process.
Okay, thank you. Thank you very
Maybe
much.
just one more quick question.
Sure. Hi. Thank you for the great talk. I have just a methodological quick question. So if you go to your result on the CNN part, where you show that the CNN can reproduce the motif, if you could go to that slide, please. Perfect. So I would like to ask you if you have some insights on, you have shown that your CNN can accurately reproduce the motif, which is a good result. But given that the motif is known, because you have Jasper and you already have all the motifs and you have all the sequences from your experiment, what is the intrinsic advantage of using the deep learning method over simply calling the motif on the sequences? Because you could use FIMO or whatever tool you prefer best to call the motifs. So what is the advantage?
Can you repeat your question?
Surely. So your CNN model, you're showing here very correctly that it learns the motifs,
Yeah.
right? So that's great. The problem is that you could have just called the motifs on the sequences, right? From the cell-free DNA. So I understand there's probably some advantage in doing this. I just wanted some insights about what such advantage is.
Just like I said in the background, at present, there is no such chip-seq experiment on cell-free DNA. That is very difficult to capture the protein binding to the DNA sequence. So I use the pre-trained model to predict the TFBS on the sequence, on the cell-free DNA sequence. That is, I cannot test the model. The TFBS predict on cell-free DNA is rarely is the true TFBS. I just can predict the potential this sequence contain or carrying the TFBS.
All right, thank you.